In this video, I'm going to talk about the biosynthesis of vitamin D. And the biosynthesis of vitamin D is very similar to that of cholesterol. And so I am not going to I'm not going to go into too much painstaking detail because particularly when it comes to things like the regulation of Hmg-CoA reductase, which, as this points out, is the enzyme that statins inhibit in order to reduce the amount of cholesterol in your blood. And so I will talk more about particularly regulatory mechanisms when we get to cholesterol biosynthesis in a later video. And so this one I'm going to focus on just what's relevant for vitamin D biosynthesis. We can see here, and so this is happening in the skin, going from acetyl-CoA, which, as I pointed out before, is from carbohydrate, and I think I may have neglected to say this in previous videos, lipid metabolism. So lipid and carbohydrate metabolism generates acetyl-CoA. Acetyl-CoA is one of those intermediates that is sort of a point at which a lot of different biosynthetic and biodegradation pathways sort of cross over. And so this can be generated from a lot of different things. And so from this, using the Hmg-CoA synthase and Hmg-CoA reductase, and a few other steps here which I will go through, we can end up with our mevalonate. So right here is the mevalonic acid, which in the deprotonated form would be mevalonate. Then we go through this squalene synthase, which is actually down here. This up here is sort of a shorthand. So squalene synthase. After some of these, in here in blue is sort of some pathways that we should be familiar with from the vitamins A, E, and K, which are these sort of prenyl phosphate synthesis pathways. So turning things like isopentenyl 5 pyrophosphate and dimethylallyl pyrophosphate into things like geranyl pyrophosphate, farnesyl pyrophosphate, geranyl pyrophosphate. These are players that we have encountered in previous videos. So this is just showing going right from mevalonate, and it's sort of just pointing out the enzyme of interest here, since these are all sort of common synthetic enzymes. It's just pointing out the one that's sort of relevant here, which is the squalene synthase to generate squalene, and then the lanosterol synthase, which is down here. And so the lanosterol synthase is these two reactions here. And so that's turning squalene into lanosterol. Lanosterol then is turned into 7D hydrocholesterol, and that happens at the 18th of these 19 steps here. And so from there, then the 7D hydrocholesterol, using sunshine, which is this UVB light here, generates the pre-vitamin D3, and then there is a shift. It calls it a sigmatrophic shift, so that's something that is spontaneous. After this is catalyzed by the light from sunshine. This will just sort of happen on its own, and it will become this vitamin D3, or cholecalciferol. And then from the skin, this can go out into the blood. So here would be the blood. This over here would be the digestive digestive tract. So this would still be the blood. And then it transports into the liver. Hepatocyte are liver cells. And so then there are these two other enzymes here that can both catalyze this reaction from vitamin D3 from the uh, calciferol into the 25-OH vitamin D3. Uh, and so that can then be transported to the kidney here. So we transported our 25-OH-D3. And then these other reactions can occur to generate things like 125-OH-D3. So these are just adding hydroxyl groups onto this vitamin D3. And so then that can also be moved here into immune cells. And this can help with things like gene signaling for genes to turn on and off and things like that. But we're pretty much going to focus mostly on this. We'll get to this step. In later videos, I will go over sort of a lot of the biological functions of vitamin D. But for now, we're just going to focus on the biosynthesis. And so, as I pointed out, this is the biosynthetic pathway here. As we can see, these geranyl pyrophosphate, farnesyl pyrophosphate, and so forth are used for several other things. Things like heme, generating heme groups, as in hemoglobin fame. And so also post-translational modifications by prenylating proteins. But let's go through some of these enzymes. So the first one is Hmg-CoA Synthase. And so that's going to generate our Hmg-CoA from 3 acetyl-CoAs. And then from there, our Hmg-CoA Reductase will turn it into mevalonate. And this just shows this is a certain type of statin here. It's written kind of small there, but this is a statin. This is showing that this can inhibit this enzyme here so that we're not producing mevalonate. And then the mevalonate can go and form the IPP and DMAP, which were these things over here, the isopentenyl pyrophosphate and the dimethylallyl pyrophosphate. So to get more into these mechanisms, so this is the mechanism for Hmg-CoA synthase. So this is the synthase. This is using the 3 acetyl-CoAs to generate the Hmg-CoA molecule. All right, so we see first that we form this covalent intermediate here with the enzyme. So this is the enzyme, and this here would be a sulfur atom. So this would likely be it would be a cysteine residue. And so that's going to bind here and kick off the CoA, and that reaction will pretty much be, well, I wouldn't say favored, because a sulfur bound to this sort of thermodynamically, it doesn't care which sulfur is bound to it, and so this will be able to happen spontaneously, is kind of the point here. And so we're losing the coenzyme-A. This shows that our sulfur on the end of it is now protonated. And so we end up with this this acetyl group here. Now it's covalently bound to our enzyme. And so this is going to deprotonate this carbon here, which we know is is able to happen because this will have this resonance stabilization here. Um, and so this will have this, the negative charge will be shared between them, but this atom right here is what will be close to this acetyl-acetyl-CoA. And acetyl-acetyl-CoA is made from 2x acetyl-acetyl-CoA, and that's why this takes 3 acetyl-CoAs, because we have this one here, and then this is an equivalent of 2 acetyl-CoAs. And this acetyl-acetyl-CoA comes in, and this will attack this carbonyl carbon that's double bound to this oxygen here, which will have that partial positive on here. And so that can attack there, and that is the condensation cleavage reaction there. And so now we have this bound here. This oxygen right here is the same as this oxygen here, and so this has been reduced into a hydroxyl group from a ketone group. So this carbon here and all of this, it comes from our initial acetyl-CoA, and then this water comes and just hydrolyzes it off of the enzyme, so that we have this hydroxymethylglutarol, or HMG, and so that's what this molecule is. And then it also has this CoA, which came in from this acetyl-acetyl-CoA. And over here, I'm just showing, so these two acetyl-CoAs come together from this beta ketothiolase to generate the acetyl-acetyl-CoA, and then this shows the acetyl-CoA coming in with this. So there's our 3 acetyl-CoAs. The Hmg-CoA synthase then forms our Hmg-CoA, and then this can go on for the vitamin D biosynthesis, but it also can have this Hmg-CoA lyase come and actually turn it into this acetoacetate species, which then can actually be turned into acetone and beta hydroxybutyrate, which are ketone bodies. So if you ever heard of the ketogenic diet, producing this beta hydroxybutyrate is the ketone body you are attempting to generate. So that's what you generate in the ketogenic diet. So this is one way that you can generate this. This can also be produced from fats, from lipids, and so that's why it's the ketogenic diet is supposed to be good for reducing body fat, because it's using fats to generate this. But once again, we'll go through that in painstaking detail in a later video when we get more into lipid metabolism and things like that. And so then the next step was our Hmg-CoA reductase, which is over here. And so Hmg-CoA reductase, because it is such a highly regulated enzyme, and because statins are used to inhibit this, this is a very highly studied enzyme. And so there's a lot to the various ways that it can be regulated as well, which, as I said, I'm not going to get into in this video, that will be saved for when I talk about cholesterol metabolism. But we see here that this protein is bound to the membrane of the endoplasmic reticulum, and it has 1, 2, 3, 4, 5, 6, 7. So it has a 7 transmembrane domain, and that is this here in the sequence. So starting at the N terminus, going to residue 339, that makes up this transmembrane domain here. Then there is this linker that links it to this catalytic domain, which is where the actual enzyme catalysis occurs. And so the catalytic domain is the only one I could find the structure for, and that's not uncommon. So transmembrane domains are kind of difficult to crystallize in order to get a crystal structure, because these are so hydrophobic in order to span the domain here, that they do not dissolve in water, and therefore they are very difficult to crystallize. And so it's not uncommon to just have on an enzyme like this to just have the structure for this section of it. And so down here we see once again this is the Hmg-CoA Synthase, which is taking the 3 acetyl-CoAs to generate our Hmg-CoA. And then our Hmg-CoA Reductase is taking this carbon right here and reducing it from a ketone into this alcohol here, this primary alcohol here. And so we see that for the Hmg-CoA reductase, we are using up 2 NADPH. And so energy is needed to form NADPH. So this is an energetic reaction. It takes energy to perform this reaction. And we see here that what's going on is this NADPH is donating a hydride ion. So that's a hydrogen with 2 electrons and a negative charge. And this hydride ion is attacking this carbon here, this partial positive carbon here, and the electrons will then jump up to there, and we end up with this mevaldyl intermediate, and so then the CoA ends up coming off of here, and we end up with this mevaldehyde intermediate. So we are losing this right here, and we end up with this aldehyde. Then another NADPH comes in with yet another hydride, and so this will come attack this partial positive carbon here. The electrons will go up, grab some hydrogen cation, and we end up generating this hydroxyl group, while these 2 hydrogens here were added as hydrides from the NADPH. And so down here we can actually see that reaction in a little more detail, or at least the active site of the reaction. And so we see we have this lysine here that is holding the carbonyl oxygen in place. Then this NADPH comes in. So this just shows it as NADH. I think the structure actually has an FAD. So I would imagine what it is that it's actually an FAD is actually doing the redox, and then this will just pass it on to the NAD, the NADPH would actually pass the electrons to the FAD, and then the FAD would pass the electrons uh, in the form of a hydride onto this carbon here. And so we see here that after we've added this hydrogen here as a hydride, that this lysine is still stabilizing this positively charged lysine is stabilizing this negatively charged oxygen. And then we have a hydrogen here on this histidine. And so this is donating that hydrogen that they show right here. And so that's where that hydrogen is coming from, is off of this histidine here. And that's allowing the coenzyme A to be kicked off of here. So the extra electrons on this oxygen are collapsing back down. Then the electrons from this bond here are going to take this hydrogen off of the histidine and remove this coenzyme A here. So we see that our molecule here, which is now in the mevaldehyde form, is still being stabilized on this lysine. And so now we have another NADPH or NADH or some electron donating molecule. NADH and NADPH are both equivalent. They're just often used in sort of different types of pathways, but they're the same. They're both just donating electrons. And so this comes in once again. We have a hydride here, negatively charged, attacking that carbon there. And then this goes up. It pulls off a hydrogen, for instance, from this lysine here, so that we generate this hydroxyl group here, and we now have our two hydrogens on here. And so I have the structure for this right here, and so we can zoom in. So this this in yellow, well, yeah, I think I said it was FAD, but that is actually an NADPH. So you can see, looking down in here, this is our nicotinamide group here. Over here is our adenine. This phosphate on the adenine is what makes it NADPH. And so this is an NADPH. So we can see here I have some of the active site residues in stick mode, and I have in gray here the histidine that we saw in our mechanism. So this histidine 866 right here is this histidine right here, and we can see it is right up here next to this. So this is the carbonyl carbon right here that is being reduced. And so we can see that our histidine is right next to it. We can see that that the nicotinamide here, which is the electron donor, is right next to it. And this lysine here in gray is the lysine that is stabilizing this oxygen right here. So we can see that the lysine nitrogen here, which would be positively charged and have 3 hydrogens, is right next to this oxygen right here. And so all of those are right next to the carbon, which is being catalyzed. And we could see in the picture, or the mechanism here, that this lysine is said to be held in here by this aspartate 755 and glutamate 767, and so we can actually see that here in this in this structure. So this right here in purple is the glutamate, and right here is the aspartate, and we can see that they are right next to this positively charged lysine side chain here sort of holding that in place there. And so this is the structure of the HMG, well, the catalytic domain of the HMG-CoA reductase. So right here is that is the most N terminal amino acid there, and if we look up here at the sequence, we see that that is number 462. And so I said up here that it's about 459. So just 3 amino acids over is where our structure begins, and that is just this catalytic domain. They do not have the linker or the membrane anchor domain in this structure. All right, and so the next step here, so the next step is, so there's 2 steps here which are just adding some phosphates onto the mevalonate, but then this step here, the mevalonate 5-pyrophosphate decarboxylase, so I have that mechanism down over here. So this is the decarboxylase. This is our mevalonate 5. This is diphosphate. You could also call it pyrophosphate. And so we see that these 2 phosphates here were added in those 2 steps before. That's just a pretty typical ATP coming in and adding its phosphate onto this molecule. So we'll just skip ahead a couple of steps here to this mevalonate 5-pyrophosphate decarboxylase. And so this is an ATP. ADP plus 1 more phosphate makes it ATP. This hydroxyl here is going to take that phosphate off of the ATP. ADP is then released. And then, so what happens here is this phosphate on here being able to leave is a, well, it's a favorable reaction because these are kind of high energy bonds, these phosphate bonds. So that leaves and allows us to generate this this carbocation here. Which carbocations are pretty unstable, so electrons from this oxygen will collapse down. This bond will be broken here as the electrons from it go to this bond here, and so this carbon and 2 oxygens, this carbon dioxide, breaks off of here, and we form this double bond right here to form our isopentenyl diphosphate or pyrophosphate, or whichever way you want to call that. And so then the next step, so there's these pentenyl, these prenyl transferase steps. We've gone over that in previous videos, but just sort of a quick overview. So this double bond here, the electrons are going to this. It's going to kick off this pyrophosphate, which, as I said, is a favorable thing to do. And so this can just happen a few times in order to go from our dimethyl allele in isopentenyl pyrophosphate to our farnesyl pyrophosphate, which is what we want for our next step here, which is the squalene synthase. The squalene synthase is well, it's not sort of, it is a 2-step reaction, but it's all happening in the same enzyme. And so we go from these 2 farnesyl pyrophosphates, which we generated from our prenyl transferase steps here, and this so this forms in what's called an epoxide. Well, actually, that's not an epoxide, is it? An epoxide would have an oxygen here, so it's just a 3 So carbons in a three-ring structure. And this is a pretty unstable structure here. These three carbon structures are are very unstable. These sort of bond angles here are not favorable, and so this is formed in it's using the kicking off of this pyrophosphate here to energetically drive that reaction. And then once again we come in and with an NAD pH to reduce this. And over here it's showing this in a little more detail. So here we have two farnesyl pyrophosphates coming in. So this actually shows the pre-squalene pyrophosphate as coming off, but that shouldn't be there because this happens all in the same enzyme here. And so this tyrosine 171 is helping this. Well, it's stabilizing this pyrophosphate and it's donating this hydrogen to it in order for the pyrophosphate to come off, which then generates this carbocation species here. The double bond on this farnesyl pyrophosphate here, the electrons for it will then attack this carbon here, and so that's how we form this covalent bond here. And then because the positive is now sort of, I guess you could think of it as being sort of transferred over, even though it's kind of hard to transfer a lack of something. But you can think about it that way. This positive charge is now transferred onto this carbon here. And so this tyrosine is now deprotonating this carbon off of here. And so the electrons, the negatively charged on here will come down and bind to the positively charged carbon here, forming this three carbon ring here. And that is our pre-squalene pyrophosphate. And then over here is the next step in that reaction. So we have this three carbon ring here. The pyrophosphate is going to come off as usual. That's a favorable thing to do. So the pyrophosphate comes off, which generates this carbocation here. We have kind of a shift as this bond shifts over to here, turning it into a four carbon ring. But then this bond here is going to move over there, generating another three carbon ring. But then we have a hydride ion coming in here from our good friend NADPH once again. And so that will attack this carbon here, which will cause the electrons in this bond to move down here. And so that's how we generate this double bond right here, as the electrons from this bond in the three carbon ring are collapsed down onto this bond here, going to the positive charge, and so that generates our squalene here. And so this is the squalene synthase. And so I have the structure for it here, and on this we can see our two farnesyl pyrophosphates in here. We have our two magnesiums here to help stabilize the pyrophosphates. This is that tyrosine 171, which is, as we said, that's going to help remove this pyrophosphate by donating its hydrogen to it. So anyway, and then this pyrophosphate will come off of here, which will allow for that 3-carbon ring to form. And so there's actually this structure for that, which if I remove these farnesyl pyrophosphates, and so this is the pre-squalene pyrophosphate here. So we can see down here our 3-carbon ring. Up here is our pyrophosphate. And so the pyrophosphate, as we saw here, the pyrophosphate will come off of here, and then we have kind of these bond movements going around on this strained ring, and so I don't have the structure for the product, but this is that intermediate, and this is that pre-squalene pyrophosphate intermediate. And so let's superimpose the farnesyl pyrophosphates. So you can see this farnesyl pyrophosphate here still is the one that maintains its pyrophosphate. This one up here is the one that will have its pyrophosphate come off, and it will bend down and bond to this other farnesyl pyrophosphate to generate the pre-squalene pyrophosphate intermediate. And then that, as I said, will go through these sort of ring shifts here to generate our squalene. And so that is the Squalene Synthase. So now that we have squalene, so up here, our squalene, we want to generate our lanosterol from that. And so that I have down here, this lanosterol synthesis. And this is kind of a fun reaction. As you can see, this lanosterol, or the squalene rather, is in the active site, bringing these carbons close together so that we can generate this reaction here, where each of these double bonds are sort of shifting their electrons over in order to generate this 4-ring structure, which is the backbone for our vitamin D and cholesterol. So lanosterol is sort of the general backbone for both of these molecules. So the first step is this squalene monooxygenase, which is actually putting this epoxide, the epoxide I was thinking about earlier when I thought that 3-carbon ring was an epoxide. But this is the epoxide. It's 2 carbons and an oxygen all in a ring. And so this is also a pretty unstable structure. And so breaking one of these bonds is going to be favorable. And so in the Cyclase reaction, first this double bond goes here to bond to here, breaking this off of here, which will become our hydroxyl here, and we are forming these rings here. So this bond is being formed. This reaction right here is forming this bond. This reaction here is forming this bond, and this reaction here is forming that bond. So we can see this sort of step by step. This double bond is attacking that carbon, which breaks one of the bonds on this unstable epoxide ring here. But because the electrons go towards this bond, we end up with this positive charge here. And so the electrons on this one are going to be attracted to that positive charge there to generate another ring, but it's also generating another positive charge. So the electrons in this double bond go to that one to form another ring, and then we end up here with this positive charge here. And then this shows that this sort of chain going here, the double bonds on this are going to attack the positive charge there, and so that will cause it to break off right here, and so that it's sort of shifting that over onto this part of the ring here, and so this it, I think I don't think that's breaking it. I think this should. I might have cut that off while copying this over. Uh, yeah, okay. So this positive charge here. So this is actually having this bond here in the ring shift down so that it's now binding to that positive carbon, but it now generates a positive charge here. The electrons in this double bond attack that and form another ring here that shifts this positive charge up to here. But now we see that that we have this hydrogen here seems to be moving up there. This hydrogen here is moving over there, and this methyl group right here that's a CH3 is moving up to there. This methyl group is moving down there, so we have a bunch of methyls sort of moving around. Now our positive charge is here. This hydrogen here is being deprotonated off of here so that the electrons collapse down onto this bond and form this double bond, and that is our lanosterol. So we form our oxidosqualene from the squalene monooxygenase. Then this squalene 2,3 epoxide or oxidosqualene cyclase generates the lanosterol. And so here is the squalene epoxidase. So this is the reaction here that is actually adding this epoxide onto the squalene there. I think this structure it doesn't have the substrate bound to it. It just has here in orange this inhibitor bound into it, and so we can't really see how the substrate binds in there. But this is the FAD here, and once again, this is the one I was thinking about earlier when I thought that the NADPH was an FAD, but this is the one where it shows an NADPH here, but the structure has this FAD. So likely what is happening is the FAD is in there, and so the NADPH is actually coming and donating its electrons to the FAD. And then there's the FAD, which is donating the electrons to this, and so there's just kind of a middleman in there, which is this FAD here. And so we can see on here, this doesn't show any of the active site residues, but we can see the active site residues I have here in purple. So this inhibitor is binding right where the substrate would also be binding. So we can see these active site residues here, and in fact I can get rid of some of the noise, and we can just look here at the active site residues. So this tyrosine here, so like I said, this doesn't show anything, any amino acids for the active site, but this tyrosine must be involved in it, and when it's in this sort of navy blue here, it must be the position it's in when there's no inhibitor, because we can see that this inhibitor gets really close, in fact so close that there would be steric clash there. And so this one down here in purple must be the inhibitor must sort of shove this down to here. I'd have to look at the paper again to know exactly, but um, that seems to be what the inhibitor is doing in the active site here. But we can see some of these other, there's a lot of hydrophobic amino acids here. We have phenylalanine, we have these two leucines here. Yeah, so those are two leucines, another phenylalanine here. We have this isoleucine right here, and we have this valine right here. So there's just a lot of hydrophobic residues going on here in the active site, which makes sense because if we look at our squalene, this is all hydrophobic. And so it would make sense for the active site to be filled with hydrophobic residues. And we can see our FAD here would come close down into the active site where this would be adding that epoxide group onto this right here. So it's adding an epoxide right here, and so that's where the reduction is taking place right there. And so we can see how that would, if we had the substrate in here, how that would be coming over the substrate in order to donate those electrons to the substrate. Let's put our cartoon structure back, and yeah, we can see that that there's, if we can kind of look down, and there's sort of this hole right here, this channel. So that'd be a hydrophobic channel. So it would be very favorable for something that's very hydrophobic, like this squalene, to go down into that channel there. And so the next structure we have here. So this is the cyclase here, and we can look down in here. This shows the product in here, which is the lanosterol. So we can see in yellow here are 4 rings on this. We can once again see that there are quite a few hydrophobic residues. We have a phenylalanine, we have some tryptophans here, we have a histidine there. And so if we look back here, so this epoxide here is grabbing a hydrogen from somewhere. We can't see in this mechanism because it doesn't show any amino acid residues, but that would be occurring down here. And so we can see that it's likely coming from this aspartate right here. So this aspartate here is, well, it would be aspartic acid when it's protonated, and that proton is likely where this hydroxyl group that's being formed here from the epoxide is gaining its proton. And so if we look back here, we see these 2 methyl groups here right next to it, which is where that epoxide was bound to it. So we would have a 3-member ring here with an oxygen. Like I said, that the hydrogen that's being donated is likely coming from that aspartate right there. And then the rest of it is sort of happening spontaneously because of that epoxide there. And so these hydrophobic side chains here are sort of holding this hydrophobic molecule in here. So once again, we can remove some of the cartoons and look at just the active site. We see we have a tryptophan here, a tryptophan here, a tryptophan here, another tryptophan over here, lots of tryptophans. We have a couple phenylalanines here, Uh, we have over here, that's a tyrosine, and another tyrosine here. So yeah, lots of hydrophobics in the active site, with the exception of this aspartate, which, as I said, is almost certainly the one that is donating the proton for that epoxide. All right, and so let's move on to the next. So now we've generated our lanosterol. So now we go through this long series of reactions. So this is cholesterol synthesis. Up here I just have the names of all the enzymes that are in for A, B, C, D, E, F, G. Um, and so we see here we start with our lanosterol. A is just adding double bonds uh, on these uh, on these carbons here. Well, it's adding one here and getting rid of one here. And then B is removing a methyl group. C is removing that other methyl group. D is now removing this double bond, which seems kind of weird since we just added it. Now it's taking it away. Uh, but uh, but evolution is a blind watchmaker, so it's not going to be like, well, I can just simplify this by not even adding that double bond. Uh, but anyway, to move on, so we've removed both of those methyl groups. We've removed this double bond. We've now moved this double bond over to here. We've now added another double bond down here. And so we could move on to make cholesterol, but we wanted to get to the 7D hydrocholesterol. That is what we will use here to make vitamin D. So I drew this big arrow going over here. So this is our 7D hydrocholesterol. And this reaction here is catalyzed by the sunlight. So this double bond moves down here, this double bond moves here, and this bond breaks. So we end up with this ring being broken here. And then that next step, which I said happens spontaneously after this, is the double bonds. So this double bond moving, oh wait, no, so this double bond is moving down here, this double bond is moving here, and this double bond is moving there. So we end up with this double bound carbon here, and the double bonds on these carbons here. And this is our cholecalciferol, our vitamin D3. So this all happened in the skin. This will then be transported to the liver, and so that is where this next step happens. So this is the cytochrome P2R1, aka vitamin D25 hydroxylase. And so this is actually using an iron bound in a heme group, heme of hemoglobin fame. And so the heme has this oxygen come down and bind to it, just as it would in hemoglobin. So now we have this oxygen bound to the iron in the heme group. This here comes and so this NADPH, this shows it reducing, but that doesn't make sense because this iron here is more oxidized. So this iron has been oxidized, but this shows it being reduced. Well, anyway, let's just kind of move on here. And so, so this has a hydrogen coming and binding to the oxygen here. We now have this peroxide intermediate here. We now have another H plus coming in, so it's going to sort of cleave this off and generate water. Now this oxygen, this shows it as a radical and positively charged. And so we have the radical reaction uh, where one electron from this bond here and one from this oxygen here are going to form this OH group on the iron here, which is now in the, well, over here it came to the fourth oxidation state. So it's in the fourth oxidation state here. So now one electron is coming from this bond here to bond to this radical on here on the 25th carbon on here, which is why it's called 25 hydroxylase, because we are hydroxylating the 25th carbon. And so that this hydroxyl group here will come off of the iron and go on to the 7D or the calciferol. Sorry, a little bit of a brain fart there. So this is the calciferol here, and we're putting this hydroxyl on it to generate the 25 vitamin D hydroxylase. So this just shows the active site, but I have the structure here, and so we can see down in here this iron in the heme group here, this big heme group in green. We see that this is being held onto here by this residue here, which is a cysteine. That's pretty common for a cysteine to hold the iron in place there in the heme group. We can look up here. This is our this looks like it's an analog molecule, but we can see that this carbon here is analogous to the 25th carbon on our excuse me, on our vitamin D3, on our calciferol. And so we see that we have this this up here where we have the carbon, and then so two methyl groups on it. And we have that going on here where we have this this carbon to carbon and then these two methyl groups right here. And we can see that that is right above this this iron in the heme group here. And so we can see how if there is a hydroxyl coming off sort of sticking out there, that would bring it right next to this group here and allow these reactions to take place. So this, as I point out here, is cholesterol 24 hydroxylase, but it's the exact same mechanism as cholesterol 25 hydroxylase, which would add the hydroxyl to that carbon there. And this structure here is the structure for the 25 cholesterol hydroxylase. There's this interesting ring structure here, which is likely something that the people who made this structure needed to add to their solution in order for this to crystallize. So that doesn't have anything to do with the function of this enzyme. But yeah, once again, we can see here this heme group, this iron is being brought close to this carbon right here, which is the analog of our carbon 25 on the vitamin D3, and so that is forming our vitamin our calcidiol here. So this is the 25 hydroxy vitamin D3. We see This has the hydroxyl on the 25th carbon here. So this was the CYP 2R1. This can also be catalyzed by the CYP27A1. These are both cytochrome P enzymes. So this happens in the liver, which generates this. This can be released into the bloodstream. It'll go to the kidney. And so this extra step can then be this extra hydroxylation, adding a hydroxyl here, can be done in the kidney, and that's just a different form of vitamin D3, which we saw up here. So this is the calcidiol that we formed from our cholecalciferol, our vitamin D3, went to the liver. We formed the 25 hydroxy vitamin D3, the calcidiol, which can then go to the kidney. And then that can be turned into the calcitriol. Yeah, and so that is the biosynthesis of vitamin D3. Just to review, we started with our acetyl-CoA and our acetyl-acetyl-CoA. We generated through Hmg-CoA synthase our Hmg-CoA. Then our Hmg-CoA reductase formed our mevalonate or mevalonic acid. We had two steps here that just sort of successively added phosphates. So it generated 5-phosphate and 5-pyrophosphate. Then we decarboxylated. Then we went into this here, which is our prenyl phosphates, things like dimethyl allyl pyrophosphate, isopentenyl 5-pyrophosphate, or geranyl pyrophosphate, farnesyl pyrophosphate, all our old friends from previous videos. But then those were used with squalene synthase to generate squalene, which then we added that epoxide group with the squalene monooxygenase that allowed us to do that cyclase, which generated our lanosterol. Then we went through that sort of long series of steps down here to go from lanosterol to our 7D hydrocholesterol, and from 7D hydrocholesterol we went here. From 7D hydrocholesterol is what is catalyzed by sunlight. So when you hear people say that, oh, go get some sun, it's good for vitamin D, this is the step they're referring to, and so that broke this ring here. Then there is this spontaneous rearrangement of the double bonds to generate the vitamin D3. The vitamin D3 is then transported from the skin to the liver, where the cytochrome P2R1, with the help of a heme, can then hydroxylate the 25th carbon on our vitamin D3, which is that carbon right there. We're adding this hydroxyl group, and so that is the biosynthesis of our vitamin D3. Yeah, I think that is everything for this. As I said, in the future, Hmg-CoA reductase will come up quite a bit more often. I'll go through a lot more of the regulation of Hmg-CoA reductase through phosphorylation and degradation and things like that. But this is the biosynthesis of vitamin 3. I hope this was enlightening, and I will see you in the next video.